Welcome back, science enthusiasts, to another deep dive. I am Hari, and I am thrilled you are tuning in today.
And I am Kimmy. Today, we are exploring the fast-paced microscopic world of enzyme kinetics. It is essentially the ultimate speed dating scene of biology.
Matchmakers is an incredibly apt way to look at it, Kimmy. Enzyme kinetics is the study of the rates of enzyme-catalyzed chemical reactions. By measuring how fast these reactions happen and seeing what happens when we tweak the environment, we can uncover the catalytic mechanism of an enzyme, its exact role in metabolism, and how drugs or modifiers might alter its speed.
Right. For a quick refresher, an enzyme, which is usually a protein molecule, acts as a biological catalyst. It facilitates and greatly accelerates a chemical reaction in the body without being permanently altered or consumed itself. The enzyme binds to a molecule called a substrate. They form an enzyme-substrate complex, transition into an enzyme product complex, and finally the enzyme releases the finished product, ready to do it all over again.
It is a beautifully elegant cycle. But it's important to note a fun, lesser-known fact from our source material. Not all biological catalysts are proteins. RNA-based catalysts, known as ribozymes, also exist and are absolutely crucial for cellular functions like RNA splicing and translation. They are composed of nucleotides rather than amino acids, but we can analyze their reaction mechanisms and kinetics using the exact same methods.
That is a fantastic point, Hari. Let us look at the general principles of how these catalysts behave. Unlike normal, uncatalyzed chemical reactions, enzyme-catalyzed reactions display something called saturation kinetics. At low substrate concentrations, the reaction rate increases linearly because the enzyme molecules are mostly free and just waiting to bump into a substrate.
But as the substrate concentration gets really high, the reaction rate asymptotically approaches a theoretical maximum. All the active sites on the enzymes are completely occupied. The speed limit at that point is simply the intrinsic turnover rate of the enzyme itself. The substrate concentration exactly midway between these two limiting cases is a very important constant denoted as Km.
Exactly. Km is basically the substrate concentration at which the reaction velocity is half of its maximum velocity, which is called Vmax. Knowing Km and Vmax helps scientists predict what an enzyme will do inside a living cell and how it will respond to changes. And speaking of foundations, we should really talk about how this is measured in the lab through procedures called enzyme assays.
Assays are fascinating. Since the enzymes aren't consumed in the reaction, laboratory procedures usually measure the changes in the concentration of either substrates or products over time. Spectrophotometric assays are super convenient because they let you measure the rate continuously by observing changes in how much light the solution absorbs as products form.
On the other hand, if you need extreme sensitivity, you can use radiometric assays, which track the incorporation or release of radioactivity over time. They are discontinuous, meaning you have to take samples out to measure them, but they can detect incredibly low levels of enzyme activity. Mass spectrometry is another powerful tool, monitoring stable isotopes to see exactly how substrates convert to products.
Here is something truly mind-bending. The most sensitive assays actually use lasers focused through a microscope to watch single enzyme molecules at work. By tracking changes in the fluorescence of cofactors or special dyes added to the protein, scientists can see the movements and dynamics of an individual enzyme, rather than just calculating the average behavior of millions of them.
I absolutely love the idea of watching a single enzyme do its job in real time. Usually, scientists focus on the initial, approximately linear part of the reaction rate, measuring it for maybe a few milliseconds to a few hours. There is even rapid liquid mixing equipment that lets you measure extremely fast pre-steady-state kinetics in less than 1 second.
Pre-steady-state kinetics is the study of those very first milliseconds before any product is fully formed and before the intermediates reach their steady-state concentration. It is vital to detect those fleeting enzyme-substrate intermediates. It is often claimed that it was actually first applied to the hydrolysis reaction catalyzed by the digestive enzyme chymotrypsin, though some sources suggest Britton Chance applied it a decade earlier to study the enzyme peroxidase.
Moving on from assays, let's talk about the big mathematical model in the room, Michaelis-Menten kinetics. Back in 1902, Victor Henri proposed a quantitative theory of enzyme kinetics, but it wasn't until 1913, after the concept of pH and buffering was introduced, that German chemist Leonor Michaelis and Dr. Maud Leonora Menten confirmed and expanded his work.
I am so glad you mentioned Dr. Maud Menten. She was an absolute pioneer conducting this groundbreaking research as a postdoctoral researcher. The Michaelis-Menten equation they developed describes how the initial reaction rate depends on the substrate concentration. It relies on a couple of crucial assumptions, primarily the quasi-steady-state assumption, which posits that the concentration of the substrate-bound enzyme changes much more slowly than the product and substrate over time.
The math can get quite deep, but a key takeaway is the concept of kcat, or the turnover number. It denotes the maximum number of enzymatic reactions catalyzed per second by a single enzyme molecule. The ratio of kcat to km measures the overall catalytic efficiency, and some enzymes are what we call kinetically perfect.
Oh, kinetically perfect enzymes are microscopic marvels. They have efficiency ratios in the range of 10 to the 8th to 10 to the 10th. They literally catalyze a reaction every single time they encounter a substrate molecule. They are operating at the absolute physical upper limit, restricted only by how fast the molecules can diffuse in water. Most average enzymes are far from this perfect, which just highlights how incredible the perfect ones are.
To visualize all this complex data, scientists used to rely heavily on linearizing the Michaelis-Menten equation, especially before computers could easily do nonlinear curve fitting. You get some great names here: the Line-Weaver-Burke plot, the Edey-Hofstee diagram, and the Haynes-Wolff plot. Scientists certainly love naming linear representations after themselves.
They really do. The Line-Weaver-Burke, or double reciprocal plot, takes the reciprocal of both sides of the equation to produce a straight line, but it actually mathematically skews the importance of measurements taken at low substrate concentrations, so modern computer software using nonlinear regression is much more accurate and preferred today. Still, they are incredibly useful for visualizing data.
Now, all of that applies mostly to single substrate reactions, but nature is rarely that simple. Multi-substrate reactions follow much more complex rate equations. If an enzyme takes two substrates, say A and B, to make two products, the mechanisms generally fall into two broad categories: ternary complex mechanisms and substituted enzyme mechanisms.
In a ternary complex mechanism, some sources suggest both substrates bind to the enzyme at the exact same time, but they actually bind sequentially to form a complex prior to catalysis. This can happen in a completely random order or a very specific ordered sequence. When you plot this data on a Line-Weaver-Burke plot and vary the concentrations, the resulting lines will intersect. DNA polymerase, which links a new nucleotide to a DNA strand, works exactly this way.
The other type is the substituted enzyme mechanism, affectionately known as the ping-pong mechanism. I just love that name. It sounds so playful. In a ping-pong mechanism, the first substrate binds, chemically modifies the enzyme, and is released as a product before the second substrate even enters the picture. The enzyme is left in an intermediate, modified state. Only then does the second substrate bind to this newly modified enzyme, react, and regenerate the original unmodified enzyme. If you plot a ping-pong mechanism, you get a set of parallel lines instead of intersecting ones. Serine proteases, like our digestive enzymes trypsin and chymotrypsin, use this ping-pong method to break down proteins.
We also have to mention enzymes that do not follow Michaelis-Menten behavior at all. For instance, multimeric enzymes often show cooperative binding. This is where the binding of one substrate molecule physically affects the binding of subsequent substrate molecules at other active sites. It is very similar to how hemoglobin binds oxygen in our blood.
Exactly. Positive cooperativity means binding the first substrate increases the enzyme's affinity for the next ones, making the enzyme highly sensitive to very small concentration changes over a narrow range. Negative cooperativity, conversely, decreases the affinity. Scientists use the Hill equation and derive a Hill coefficient to quantitatively measure this degree of cooperativity.
So we know how enzymes speed things up and work together, but how do we slow them down? Enzyme inhibitors are molecules that reduce or abolish enzyme activity, while activators do the opposite. Inhibitors can be reversible or irreversible. Reversible ones are traditionally grouped into competitive, uncompetitive, and non-competitive inhibitors based on their effects on Km and Vmax.
In competitive inhibition, the inhibitor binds directly to the active site, literally competing with the substrate. This increases the Km, meaning you need more substrate to reach the same rate, but the maximum velocity remains exactly the same. Non-competitive inhibitors bind to an allosteric site elsewhere on the enzyme. They do not change the binding affinity for the substrate, but they do decrease the overall maximum speed.
Then you have irreversible inhibitors, which usually covalently modify the active site residues, permanently shutting the enzyme down. There is a rather dramatic term for a specific type of these: suicide substrates. These follow a mechanism-based inhibition, where the enzyme binds the inhibitor and actually transforms it into a highly reactive group that irreversibly destroys the enzyme's own function.
It is tragic but fascinating chemistry. To uncover these specific chemical mechanisms and figure out the exact sequence of steps, scientists use clever tricks like the primary kinetic isotope effect. By replacing a hydrogen atom with its heavier, stable isotope, deuterium, they can determine if the breaking of that specific bond is the rate-determining step, simply because bonds to deuterium are physically harder to break.
They also track stable isotopes like oxygen-18 to see exactly where individual atoms end up in the final product. All of this kinetic data helps confirm the induced fit model of enzymes, where the initial weak interaction between the enzyme and substrate rapidly induces a conformational change that strengthens the binding and aligns the catalytic residues perfectly for the reaction.
And if we really want to blow some minds before we sign off, we have to mention quantum tunneling. Some enzymes accelerate hydrogen transfer reactions by allowing particles like protons or electrons to pass straight through an energy barrier rather than having to climb over it, entirely due to their wave-like quantum nature.
Quantum biology at work in our very cells. From ping-pong mechanisms to quantum tunneling, enzyme kinetics is a true testament to the incredible, invisible complexity keeping us alive every single second of the day.
It really is breathtaking when you think about it. Thank you so much for joining us on this molecular journey today. We hope you learned something entirely new and exciting. Be sure to tune in next time for more deep dives. Until then, stay curious, keep exploring, and have a wonderful day.
This episode is based on the Wikipedia article Enzyme Kinetics, which is available under the Common Rights Attribution Share-alike license. This podcast was crafted using artificial intelligence.